this is Dr. Cassandra Quave. In this lecture, we'll be covering remedial plants used in the treatment of cancer. This lecture is offered through Emory University's course in Botanical Medicine and Health. The learning objectives for this lecture are to understand what is the link between diet and cancer, and why do some edible plants have protective effects against cancer. What are some of the main anti-cancer drugs or therapeutic agents that are derived from plants? What are their historical uses in traditional medicine? The types of cancer they are used to treat, and the basic concepts behind their mechanism of action. To begin this lecture, I wanted to start off with a slide that highlights 3 of the botanical stars in this realm. And on the left, I want to highlight Catharanthus roseus, also known as the Madagascar periwinkle. This important plant is a source of Vincristine Vinblastine. Taxus brevifolia, shown in the middle, is also known as the Pacific yew, and this is the original source of Taxol. Camptotheca acuminata, on the right-hand side, is also commonly known as the Chinese happy tree, and this is a source of Camptothecin. Now, before I dive in to the details on each of these plants, we're going to cover their chemistry, the mechanistic basis of their activity, and the different types of cancer they're used to treat. First, I wanted to give a little bit of background on how some of these different drugs were discovered through the National Cancer Institute's program for drug discovery and cancer therapeutics. So the National Cancer Institute had a precursor program known as the Cancer Chemotherapy National Service Center. This later became known as the DTP, or Developmental Therapeutics Program. The objective of the organization was to obtain compounds, screen and evaluate them in preclinical and then later in a clinical setting. And this research was focused mainly on cancer and HIV and AIDS drug discovery efforts. Now, before the 1960s, there was a big focus on screening microbes, but the study of and things like they screened they were screening natural products from the fermentation of microbes, and there really wasn't that heavy of an interest in searching for drugs from plants until the 1960s, when studies on the Madagascar periwinkle and also on the May apple instigated a further interest in medicinal plants. And at that point in the 1960s, only about 1,500 plants were really looked at in these screening efforts. So between 1960 and 1982, about 114,000 extracts coming from 35,000 plants were screened, and there was a massive collection effort that was undertaken. Plants were brought in from over 60 different countries, and some of the major discoveries from these efforts included Taxol and Camptothecin. Now, I suppose that we really need to be able to define what cancer is before you can really understand how to treat it. So in general, cancer can be defined as a class of diseases in which a group of cells display these unique characteristics, one of which is that of uncontrolled growth or division beyond the normal limits of typical cell growth. Invasion, this means that the cells intrude on and destroy adjacent tissues, and sometimes you can also have metastasis, in which you get a spreading of cancer to other locations in the body through either the lymph fluids or blood. Now, in this lecture, we're going to be focusing on just 6 important plants, 2 in particular for prevention, cancer prevention, and 4 in therapy. However, I wanted to make it clear that there are many, many more plants currently under investigation now for both the prevention and treatment of diverse cancers. Now, as I start thinking about how to teach you about cancer prevention, you really need to be introduced to some basic concepts of what are some of the things that can lead to cancer, and how can we mitigate those through prevention efforts. So I want to start with the definition of an antioxidant. Antioxidant has become kind of a buzzword in the popular media now, with lots of different drink and beverage products and other types of food formulations that are touted and advertised as being potent antioxidant formulas. Well, many people, unfortunately, don't really understand what an antioxidant is or why we need to have these in our diet. So I'll give you some groundwork here to work with. So an antioxidant is basically a molecule that is capable of inhibiting the oxidation of other molecules. And what is oxidation? Well, oxidation is a chemical reaction that transfers electrons or hydrogen from a substance to an oxidizing agent. This sounds a little bit confusing, but we'll show you a picture in the next slide that should help to clear this up. The basic thing that you need to know here is that oxidation reactions can produce free radicals. In fact, production of free radicals is a part of the normal process of our respiratory pathways. Whenever we go in the mitochondria and energy processing, you have release of these free radicals, which can then cause a chain reaction in the body. There are other things that you can do to your body to increase the number of free radicals that circulate. This includes things like exposure to ultraviolet light, or getting in too much sun, or being in a tanning bed, for example, cigarette smoking, exposure to other kinds of environmental toxins. All of these are related to the production of free radicals. So I mentioned that free radicals can cause a chain reaction. What happens when this chain reaction occurs in the cell, it can cause damage or death to the cell. It can also cause mutations to occur in the DNA, and then downstream effects can lead to cancer. Now, the role of an antioxidant is that it can terminate these chain reactions by removing the free radical intermediates, or neutralizing the situation, and inhibiting then that chain reaction of other oxidation reactions. And they do this by being oxidized themselves. So oftentimes you'll find that antioxidants are often reducing agents like your thiols, ascorbic acid, or your polyphenols. Plants like your berries and that are rich in polyphenols are excellent sources of antioxidants. Other types of antioxidant compounds include things like vitamin C or ascorbic acid, your carotene compounds, and also your alpha tocopherol or other tocotriene compounds. These are vitamin E and vitamin E derivatives. Now, how does oxidative damage lead to cancer? Well, what happens is that it can actually cause mutations in the DNA, and again, increase the risk of cancer susceptibility. Now, our body not only can use exogenous antioxidants. That means exogenous refers to antioxidants coming from outside of the body, such as through the diet. But we also produce our own endogenous antioxidants. And these include things like superoxide dismutase, catalase, glutathione peroxidase, glutathione reductase, and glutathione S-transferase. And these help to, again, stop that chain reaction and protect DNA from oxidative stress. In industrial settings, antioxidants are also applied to food as an additive to help guard against food deterioration, because also food products, like any other biological product, is also at risk for oxidative damage and deterioration. So here, if you look at this picture of what is an antioxidant, I mentioned in the last slide that here you have a normal oxygen atom, and on the upper left hand corner you can see that what happens sometimes during normal processes in your body's energy metabolism is that you can lose an electron circulating around this oxygen atom. And what happens then is you get a chain reaction, right, where free radicals can hit cell to cell they're trying to find that electron to go back and fill that gap that's missing in their orbit. But instead of, what happens is they go and they steal these electrons from other cells, which can then cause problems in those cells, and then you get the chain reaction because that next atom that gets affected, then that starts searching for a replacement electron. So the way that antioxidants work is that they actually have parts that they can donate to the unstable free radical, and they can do so without becoming a part of the chain reaction themselves. Now, speaking of antioxidants, let's start by looking at some of our cancer preventative agents. The first I'm going to cover here is Camellia sinensis. Camellia sinensis is in the Theaceae family, and this is also known as green tea or black tea. Now, epicatechin derivatives, and these can include polyphenols like aflavins and thiorubigens, that are found in green and black tea can show very strong anti-carcinogenic effects. And this has been shown in laboratory studies that they exert antioxidant and free radical scavenging activity. And what I mean by free radical scavenging activity is, again, compounds like this on the right can go and neutralize those free radicals and stop the chain reaction in the body. They also are active in inhibiting protein kinase C and in inhibiting cellular proliferation. These are important processes in cancer, and they have anti-inflammatory properties. Now, tea leaves are, of course, central in traditional Chinese medicine and are used in the treatment of asthma and also in cardiovascular diseases. And you can see here one of the important compounds derived from tea. This is epigallocatechin 3-gallate. The next plant that I wanted to highlight is that of turmeric. Now, turmeric is from the, also known as curcuma longa is the scientific name, is actually a plant from the ginger family. It's in the Zingiberaceae family, so this is a relative of ginger. One of the major compounds is that of curcumin, and you can see the structure here at the top. Now, turmeric has been used in complementary and alternative medical therapies for its anti-inflammatory activity, and it's also very important in Ayurvedic medicine. The plant is native to South Asia, and the rhizome is actually dried and made into a powder that is then used as a spice in traditional Indian cooking and other types of South Asian cooking. And it's also used for dyeing and for giving food a nice color. And you can see here on the right an example of some turmeric powder. I like to throw a little bit of turmeric even into my own dishes. Often when I make rice, I throw a little in there and it looks a little bit like yellow rice, and I'm giving myself a nice boost of some interesting anti-mutagenic and anti-carcinogenic antioxidant kind of properties of the curcumin. Now, curcumin is found in both curcuma species and Acorus calamus, which is another species. It has, again, these anti-mutagenic and anti-carcinogenic properties. That means that it helps prevent mutations in cancer pathways. And specifically, it can inhibit pre-neoplastic lesions of the breast, colon, and neoplastic lesions of the skin, stomach, duodenum, and the colon. It also works by suppressing phosphokinase C. Now, what's interesting is that there is work underway currently at Emory in a lab that focuses on curcumin mimics. These are compounds that are designed to mimic, or they're slightly different from the original natural product. And they're looking at the many different applications of or possible applications of these curcumin mimics as medicinals. And you can learn more about that by following this web link here, listed at the bottom of the page. Okay, now let's begin our talk about some of the important medicinals that have been developed in the treatment of cancer. And the first I want to begin with is the Chinese happy tree, also known as Camptotheca acuminata. Now, Camptotheca acuminata is the source of a compound known as camptothecin, and this is an alkaloid that was discovered in the 1957 National Cancer Institute screen of woods and barks. And it works in a unique way. It works by inhibition of topoisomerase 1, and this basically prevents DNA unwinding. So it has activity against leukemia, but it also has some detrimental toxic side effects. There have been 2 products that have evolved from research on camptothecin, and this include irinotecan and topotecan. And these are both semi-synthetic derivatives of camptothecin. And again, this is a common story that you're going to see over and over again with many botanical remedies in which scientists have used the natural product that's originally derived from a plant and then taking that basic structure, that very complex, unique natural product structure, and then using molecular modeling techniques to kind of tweak the compound to improve issues that they've run into with regards to solubility or bioavailability, toxicity, and so on. So let's say that you find an active compound but it's not very soluble, you can tweak it, add or subtract some things to the compound and hopefully improve its solubility and thus ability to be delivered to the patient. Now here you can see that how it was used as a template. In the case of irinotecan, this has been marketed as Camptosar or Campto. It is less toxic than camptothecan, and it was approved in 1994 in the U.S. to treat metastatic colorectal cancer. It's also effective against lung cancer and some leukemias. Topotecan, on the other hand, is marketed as Hycamentin, excuse the pronunciation, and it was approved in 1996 in the U.S. to treat ovarian cancer. So again, 2 very important drugs that came from, that were developed based on the structure of a natural product, in this case that of the Chinese happy tree. Another really great success story is that of Vincristine and Vinblastine. These are 2 compounds that were isolated from Catharanthus roseus, also known as the Madagascar periwinkle. Now, Catharanthus roseus has a very interesting long history in traditional medicine. In Europe, it was used as a diabetes remedy. In China, it's been used as an astringent, and for its diuretic properties, it's also used as a cough remedy. In the Caribbean, it's been used for eye infections and diabetes. It also has this interesting reputation as a magical plant. In fact, Europeans thought it could ward off evil spirits, and the French have been known to call it the violet of the sorcerers. Now, this is the source of the vinca alkaloids. They're called the vinca alkaloids because previously this genus, this plant was identified under the genus Vinca rather than Catharanthus, but the current taxonomy is that of Catharanthus. Now, there was an Eli Lilly screening program that first identified the activity of this plant in treating certain cancer cells. Overall, there were more than 140 alkaloids identified from this plant, and mostly they were the indole alkaloid types, again called the Vinca alkaloids, based on that old name of the plant, which was Vinca rosea. Now, of course, Catharanthus roseus. So how do these compounds work? The mechanism of action is that they inhibit mitosis by binding to tubulin, and this prevents the cells from making the spindles needed to move the chromosomes during cell division. So if you flash back to looking at cell division from perhaps your high school biology class even, If you remember the formation of spindles as a cell divides, this again helps preventing those cells from making the spindles to move the chromosomes, thus stopping cell division, in this case cell division of cancerous cells. Now, vinblastine is marketed as Velby by Eli Lilly. It's been used in the treatment of Hodgkin's disease, lymphoma, advanced testicular cancer, advanced breast cancer, and Kaposi sarcoma. Vincristine is marketed as Oncovin by Eli Lilly, and it's been used in the treatment of acute leukemia, Hodgkin's disease, and other lymphomas. There are also some semi-synthetic derivatives on the market. There is Vindicine, which is marketed as Eldocine, and this is used to treat leukemia and lung cancers, and Vinorelbine, which is marketed as Navalbine, used to treat ovarian cancer. Now, one issue with vincristine is that it has a very low natural yield. It's only 0.002% for vincristine, so it can be very expensive to make. However, the fact that these are dimeric indole alkaloids are also very difficult to synthesize. So there are some issues in trying to synthesize these in the lab. And you can see from the complexity of their structure why that would be so. Okay, another great story is that of Paclitaxel. So this is the cancer drug coming from the Pacific yew, or Taxus brevifolia, in the Taxaceae family. This is a shrub that grows in the Pacific Northwest. Paclitaxel is actually a taxane diterpene. Again, another one of those compounds discovered in the 1960s NCI drug screen. And what was interesting is that it showed strong activity against solid tumors, against melanoma, and leukemia models. And what's most interesting about this is that a new mechanism of action for cancer treatment was actually discovered with this drug. And this discovery was by Susan Horwitz's group. Basically, Taxol can inhibit mitosis, but it also does so while stabilizing the microtubules, and it can inhibit depolymerization back to tubulin. So this was a really important discovery, also just on the basis of this the mechanism of action. However, with Paclitaxel, there was a huge problem. There was, again, a very low yield in the plant of this compound in the plant. So originally they were taking it from the bark of the Pacific yew. The solution that was found was to do a semi-synthesis procedure of Taxol by converting metabolites that were available in a larger quantity in the needles of the English yew, or Taxus baccata. This is another species within the same genus. And what was great about this innovation is that the needles were a renewable source. It was a more sustainable method of harvesting. There was no more need to actually kill the tree by removing the bark in this case. And here's a picture of Taxus baccata, or the English yew. Now, Taxol by Bristol Myers Squibb was approved in the U.S. in 1993, and it was used for treatment of ovarian cancer and secondary treatment for breast and non-small cell lung cancers. Docetaxel was approved in 1995 and marketed as Taxotere. This was a more water-soluble version of Taxol, and it was used in the treatment of breast and ovarian cancer. Abraxane is Paclitaxel bound to albumin. Again, just taking that same compound and tweaking it with some things. This was approved in 2005 and is used in the treatment of breast cancer that is unresponsive to other forms of chemotherapy. Okay, the next plant that we're going to discuss is that of Podophyllin peltatum. This is in the Beriberidaceae family. It's commonly known as the Mayapple. Now, the interesting thing about this plant is that it has a very long history of traditional use of the rhizomes as a medicine by Native Americans, in particular by the Penobscot Indians of Maine. Now, they would take the plant that was dried and made into a powder and then eat or drink this powder as a laxative or as an antihelmintic. So this is in the treatment of intestinal worms. What was also very interesting is they would make a poultice of the powder, which was then applied to warts and skin growths. Now, anytime you have a plant that's being used to treat things that are related to uncontrolled cell division, like in a skin growth or wart, it kind of sets off alarms that, oh wow, this may be an interesting plant to study for potential anti-cancer properties. Now, currently, plant extracts of this are used as a topical treatment, again, for warts and skin growth, and the compound here is podophyllin. Now, podophyllotoxin lignans work by binding to tubulin, and these are the members of the spindle poison group of agents. They work by preventing microtubule formation. Podophylox is actually a purified form of podophyllin that acts as a poison against cells that are undergoing mitosis. So this is not something that is useful for systemic applications, but it can be used topically in genital wart creams. There are also some synthetic derivatives of podophyllin. These include etoposide, which is marketed as Vepicid. This is used in the treatment of small cell lung cancer and testicular cancer and lymphomas. Teniposide is marketed as Vumon, and it's been used in the treatment of brain tumors and in childhood acute leukemia. And again, I want to point out the interesting chemistry of these two semi-synthetic derivatives at the bottom of the page. So to wrap up this lecture, I just wanted to again reemphasize the role that medicinal plants have played in some of our major cancer drugs still in use today. The important work that the National Cancer Institute conducted was, of course, critical to these developments, as well as the work of other private pharmaceutical companies. Overall, they've tested more than 35,000 plant species through this process for anti-cancer activity in the past century. And during this process, a few very important drugs have emerged, including Taxol, Vincristine, and Vinblastine, which are all used today. In all these cases, again, traditional medicine gave some clues, and there's still much more work to be done. I don't know that, I think that more of a focus on, again, on the ethnobotanical approach to drug discovery, looking and focusing again on plants used in traditional treatments for things like skin growths and warts could be very fruitful in identifying new possible leads for anti-cancer medications. So this is all I have for today's lecture. Thank you for your attention, and stay tuned for more posts.